In this video, I'm going to talk a bit about how taste works. And some of you may be familiar with this image right here of the tongue having different regions that are sort of used for different types of taste. But this view of how taste works is actually a myth. And so that's why it's crossed out. So what's actually true is that the taste buds, which we can see here, then we zoom in, the taste bud is this right here, and we have these papilla, so there are these regions in between here. And if we zoom in on one of these little spots here, we actually look at what the taste bud looks like. And so it contains a bunch of different cells, well, three different cells to be precise. And every taste bud contains all three of those cells. So every taste bud can sense every kind of taste. And so we see that there are the three different cells in there, and the three different cells are right here. And so the first type is this glia-like cell. And so it's glial-like because it doesn't actually interact with neurons. And so it's thought to play kind of a support role. I have seen some sources that say that it might be responsible for salt taste in some way, but whether that's true or not is still up in the air. And in fact, if we look over here at type 3, this says that it is responsible for sour and salty. And both sour and salty, and this might seem strange, are still quite not well understood. Even, in December of 2021, when I'm recording this, they're still not well understood. So the type 2, though, which is involved in sensing sweet, umami, and bitter, that one is a bit more well understood. It uses a mechanism called the Gproteincoupled receptor, which I'll get into in a little while, to sense the taste, to sense the presence of sugars and the monosodium glutamate of umami and the various molecules that are bitter, where the sour and salty actually use ion channels. So we'll find that these use ion channels to actually sense the presence of either sodium ions, which is salt, or hydrogen ions, which is sour. And so hydrogen ions is the Bronsted-Lowry, so the Bl acid. And so that's why acids, things like citric acid and vinegar and things like that are known to have a sour taste because we sense sour through these H plus ions, through these hydrogen ions. And so we have our taste bud here, and it has these three different types of taste buds. So there's the circumvallate, the foliate, and the fungiform, which connect to different types of nerves. So there's the chorda tympani, there's the glossopharyngeal, and there is the down here the vagus nerve. And so those bring the signal from the tongue to the brain, to the gustatory cortex. So gustation being sort of the fancy word for taste. And so the nucleus of the solitary tract and the thalamus and things like that. So however consciousness works, that's what brings it into our sort of conscious awareness of taste. And so the different cells can have the different types of receptors in them, and I'll go over the receptors here in a little bit. So there are a few ways that the taste can be sensed. So there is this labeled line coding, where each cell expresses just a certain type of receptor for sensing that taste. So maybe cell 1 has the bitter tasting receptors, and so that will taste the quinine, which is bitter. Cell 2 tastes the sugars, so sweet, and then cell 3 has receptors for the MSG or umami. And then this is showing that population coding, so multiple different cells would have these, and so that would be responsible for the salty and sour taste. All right, and so now if we move down here to look a little bit closer at these cell types, so I wanted to read this from one of the papers, and as usual, links to all the sources will be in the description. So the type 2 cells are the receptor cells for the taste qualities of sweet, bitter, and umami, mediated by the taste receptor family of Gprotein-coupled taste receptors and regulated by phospholipase C. So the points of contact between the type 2 cells and nerve fibers often exhibit a non-conventional specialization involving subsurface cisternae and atypical mitochondria. So no conventional synapses. It does not contain conventional synapses. It does not contain, the usual presynaptic vesicles and postsynaptic membrane thickening with the type 2, whereas the type 3 do not express the G protein coupled receptors, but do form conventional synapses with the afferent nerve. So afferent nerve being the type of nerve that brings signals from the body to the brain. Efferent nerve, so essentially just replacing that A with an E, is what brings signals from the brain to the body. So this is talking about bringing signals, from the taste buds to the brain. And so these ones do use a normal or conventional type of synapse. All right, so down here, this is looking at these cells in a little bit more detail. So remember the type 1 is that glial type, and this shows that it has this NTPDase, which breaks down, nucleotide triphosphates. And what we'll see here is, and I have it boxed in green here, that the bittersweet and umami stimuli type 2 cells release ATP via non-vesicular release channels. And so it's ATP that's actually being used as the neurotransmitter in this case. And so I actually have this image here on the right, and this goes through both the ionic stimuli and the sort of sweet, bitter umami stimuli. And so it kind of shows both of them in the same cell, but they would be taking place in the type 2 for the complex stimulus and type 3 for the ionic stimuli. But the ionic channel, so ions move in through this channel here, and it causes depolarization, which causes an action potential, which then signals for the release of the ATP. Whereas in this one we have the G protein coupled receptor, which I'll get into in a little bit. So the second messengers, which then releases calcium from the endoplasmic reticulum. And this does show a vesicular release, but as I said over here, the ATP is released from a non-vesicular, from channels, from just, like a channel, a protein channel that releases the ATP into the synapse, which is then picked up by the afferent neuron, and the signal is then brought to the brain. So here I'll talk a little bit about how action potentials work. And so what we have here first is this. So this is the resting membrane potential. And so on the inside of the cell, we have these potassium ions. These A's with the multiple negatives are negatively charged proteins. And so then on the outside here we have sodium ions. And so we have this charge separation that creates a potentially a charge gradient where it's more positively charged on the outside and more negatively charged on the inside. And we have these proteins embedded in the membrane that actually pump potassium inside and pump sodium outside in an ATP dependent way because it uses energy to move things sort of up their gradient. But then what happens when we have an action potential is the sodium, there will be what are called gated channels. So there can be ligand gated channels and charge gated channels. So the ligand gated channels are what the neurotransmitters bind to. It opens it up and it allows sodium to start entering into the cell. So the sodium will enter and the inside of the cell will go from this this negative 70 millivolt voltage. It will depolarize as sodium moves in, and that causes the action potential. And then repolarization happens as potassium moves out. We actually end up with this refractory period of a hyperpolarization, and then the pumps have to repump the potassium back in and the sodium back out. And so this can move down the cell. And so what we have here is at the resting potential, it's negative inside and net positive outside. We have the sodium move in right here, which causes the outside to become more negatively charged, the inside to become more positively charged. And so we see right here that we actually move above the zero here into this positively charged on the inside right there. And so what happens is we end up with positives sort of a little bit downstream of where that is, which then activates charge-gated channels down here, which then causes more sodium to move in down here. And so this action potential moves along in this fashion. Then we see behind it we have the potassium being pumped back out to sort of repolarize things. And in fact, we get this period of hyperpolarization here as the potassium moves back out. And then, of course, the potassium will start getting pumped back in and the sodium get pumped back out so that we can sort of go back to what we had right here and sort of back to that resting potential, which was in this state right here. And so that is what is going on here, where it shows this depolarization. So both of these, the complex stimuli, so the sweet, umami, bitter, and then the ionic stimuli, the sour and the salty, both result in this action potential. We see that this has the sodium being pumped in, the potassium being pumped out, and then over here this shows the calcium being pumped in. So calcium is pumped in either at the synapse, or we have the calcium coming from the endoplasmic reticulum over here. And so calcium is acting as that second messenger. And so over here, these are the so this is the channels used. So this is the well, this shows a serotonin receptor, but these are actually the ATP receptors on the afferent nerves. And so what we see here, so this is sort of a cross section of the protein. And so in the open state, which is when ATP is bound, and we can actually see ATP bound in here, we end up with this open state. And so the ions, which are right here, so we would have maybe sodium ions here. So sodium can actually move in and move all the way through the channel where the plasma membrane would be right around there. And so it can actually move those charged ions through the hydrophobic channel. So this is the apo state, which means that there is no ATP bound. And we see we have these gates closed here. And we also have a desensitized state. So if ATP sort of stays bound just sort of all the time, you end up desensitizing the receptor, and we close this gate so ions can no longer move all the way through here. And so this is just looking at sort of the full structure here, and this is actually a trimer here. So there's actually three different proteins sort of coming together to form this channel, and you can actually see, you know, there's an ATP there, an ATP there, and you can kind of see the one in the back there. So each of those proteins have an ATP bound. You can see it better at the top view here, where we have 1, 2, 3 proteins. And then we can look at this. This is that ion channel right here. And I actually have the structure here. So this only shows one of the proteins in the homotrimer here. But what we can see is we have in yellow here, this is ATP. We have this ATP bound right here. And so what we have down here, so in green, that is in the open state, and in the cyan, that is in the apo or closed state. And so we can see that there is a shift down here in the conformation, where up here we can see that the cyan and green overlap almost perfectly. So down here, so when that ATP binds, it generates a conformational change that propagates through the protein and causes this part of the protein to sort of move out of the way and open that gate and allow ions to move through. And so that is so it's called the P2X3 protein. So that is what we have shown. Well, I don't think it shows it here, but it is the P2X3 protein, which is the receptor on the afferent nerve. So this protein is in the nerve, not in the actual taste cells. And so now we'll talk a bit about the receptors that are actually present in the taste cells. And so what we have over here for the umami, the sweet, and the bitter are these Gprotein-coupled receptors. So bitter, it says there are somewhere around 30 different types of receptors. So there's essentially a lot of ways to taste bitterness. Sweet, we have this T1R2 plus T1R3 heterodimer. And then umami, we have this T1R1 and T1R3 heterodimer. So both the sweet and umami use the T1R1 or T1R3 rather as part of their heterodimers. Then for sodium, we have this epithelial sodium channel, shortened as ENAC or ENAC, as you might pronounce it. So then this shows for sour this PKD2L1, but there is a bit of controversy about which receptor is actually responsible for the sour taste. So there is some recent papers that show that a receptor called OTOP1 is actually responsible. And so it's either one of these, and what we'll see interestingly is that both of them sort of are proposed the same sort of mechanism of action, whether it is the PKD2L1 or the OTOP3, whether it's either one of those proton channels, they both are proposed to use the same mechanism of action. All right, and so the first one, so I'm only going to talk about the salt, the sour, and then the sweet ones, because the umami, sweet, and bitter all use the same sort of mechanism of action, so there's no point in sort of going over the mechanism 3 different times for 3 different tastes. But the first one I'll talk about here is the salt. And so so I'll kind of read off this and go over the figure. So this work leads, this is from a paper, and so salt, like I said, just like with sour, it's still not really that well understood how it actually works, which just kind of seems crazy to me that nobody's figured that out yet. But this is from a 2020 paper, and they are proposing a mechanism based on, a bunch of experiments they ran. And so their work leads to a model for a salty taste in which sodium ions in the oral cavity enter taste receptors through the ENaC, the epithelial sodium channel, leading to membrane depolarization. So that's what's going on up here. So we have our sodium here moving in through this ENaC receptor here up at the apical end of the cell, which will result in a action potential that will move down through the cell, so down to here. And we see this sort of purple thing right here that is the afferent nerve. So the action potential moves down there, so it generates an action potential. Then this CALM1 and CALM3 channels open in response to the depolarization and release ATP onto the afferent nerves. So that's what they're showing right here. They have this CALM1 channel right here. This ATP moves out and binds to the P2X2 and P2X3, which was that receptor I was talking about just a little bit ago on the afferent nerves. And so it binds to that, and then that allows sodium to move into the afferent nerve and generates an action potential in the afferent nerve. And so the salty cells can be uniquely identified by co-expression of CALM1 or CALM3 and the ENaC alpha, both of which are required for the amiloride sensitive salty transduction. So amiloride, which is this molecule down here, is an ENaC inhibitor, and so that inhibits the ability to taste salt. And what they're saying here is that these salty cells, or the cells that taste salt, are uniquely identified by cells that express both this channel here, the CALM channel, shown right here, and the epithelial sodium channel, which is shown up here. So it has to have both of those in order to allow for the taste of salt, and that is sort of the evidence that they're using for their model of salt taste. And so, like I said, this is from a 2020 paper, so this is still pretty new. There's still work going on in this, so it's not completely settled how this actually works. And what we will find is that is the same for the sour taste. And so I actually have the OTOP1 and the PKD2L1 mechanisms here. And so this is sort of the simplified version where OTOP1 allows hydrogen to move in through this channel, which has a change in pH, and then that actually inhibits these potassium channels that allow potassium to move out, which is what the cell would do in order to sort of stabilize the charge. And this causes a depolarization, which results in an action potential, which results in the calcium influx here, which acts as a second messenger that then releases the ATP. And so that's kind of also what we're showing up here. We have the H plus going in. It's inhibiting, that's what the flat bar right there means. It's inhibiting the potassium outflow. We end up with the action potential moving down here, then the calcium influx, and then the nucleotide, the ATP release. And so that is sort of the OTOP1 sort of mechanism here. And so, like I said, the PKD2L1 is pretty much the same thing. So this is actually showing a weak acid sort of being moved in over here. And so we have our conjugate base here with our H plus, and when those two join together, it neutralizes the charge on it and allows it to move in through the cell membrane. Then we have the H plus coming off, and this shows that we can also have the H plus being moved in through this channel here, which then inhibits the potassium channel and causes action potentials here. And so this is just showing for the strong acid, which would be always dissociated, that this would just move in through that channel. And so this is showing that in non-sour taste cells, there are too many of these potassium channels here for the action potential to actually be generated. What they showed in their paper is that the sour taste cells had a lot fewer of these potassium channels in them. And in fact, in non-sour taste cells, when they did a knockout of these, or a knockdown rather, reducing the number of potassium channels, they found that they could induce action potentials in the non-sour taste cells in the presence of acids. And so that is sort of the evidence they were showing for their model using the PKD2L1 ion channels. So I think, I mean, both of these papers that use these figures are from fairly recently. I think maybe 2018 or 2019. So once again, this is still an area of active research. So yeah, this is the salt channel, and I do have the structure for the epithelial sodium channel right here. So you can see this is the trimer, where we have this this sort of purplish part here, this sort of salmon part here, and this magenta part here as the three different receptors. And it's actually a heterotrimer because there are three different proteins that actually come together. There's the alpha, beta, and gamma, and this actually shows all three of them. And it's the alpha, which if we move in here, you can see this sodium ion here, which when bound into the alpha monomer actually opens up the channel. And so we're looking down, you can see sort of in the middle here this opening, which is where the ions would actually move down through there. And so you can actually see how the structure would work for that. This part down here is the transmembrane domain. So this is where the membrane would be. This part would be outside the cell. And so when there's sodium outside the cell, it binds in here, opens that up, and allows sodium to move down in through here and into the cell, and then generating that action potential. And then so I have here, this is the OTOP1 receptor, and we see that this is actually a homodimer, And we can see all these sort of multicolored chains down here. Those are all cholesterol molecules. And so this is sort of innervated with a bunch of cholesterol molecules in this protein. And that makes sense because the membrane would be right here. And so you would want those sort of hydrophobic things going on there, those hydrophobic molecules helping to hold it into the membrane. Then I have over here, this is one of the ion channels, and I have all these amino acids shown as sticks. Those are all polar amino acids, and so it creates a polar environment here, which would allow ions to move down through there. So that would allow the hydrogen ions, the acid ions to move down. So up here would be outside the cell, so the ions could move down through that polar channel and into the cell. And so this is the OTEP1. This is the OTEP1 channel right here. So that was, as I said, just one of the models for this. So it's either the OTEP1 or the PKD2L1. And in fact, in the paper where I got these figures, the OTOP1 actually talked about how if they sort of knock out the OTOP1, there is still a small amount of action potentials going on, and they propose that that might possibly be this PKD2L1. And so it's possible that both of these mechanisms are actually at play in the sour taste. All right, and so now we will move down here to sweet taste. And so I have this image of the two structures here of the T1R2 and T1R3 receptors. And we can see where the sucrose, which is table sugar, and glucose are bound sort of in here. We can see where aspartame and several other of these sort of fake sugars actually bind onto these artificial sugars bind onto these receptors. And we can see down here, this is sort of a cartoon figure of it. And we see we have these ligand binding domain 1 and 2, ligand binding domain 1 and 2. And these use this, what they call the Venus flytrap model here, where the taste substance kind of goes into this sort of Pac-Man mouth like that. And I actually have the structure of the Pac-Man part, so just this part here. So I have the structure of that right here, and I actually colored them kind of the same as what was on that image there. So this purple one here and this cyan one here correspond to the T1R2 and the T1R3, and so we can look at that here. And I shaded the sort of top jaw and bottom jaw of these Pac-Mans, I guess, here differently, so we can kind of see that this top and bottom jaw could kind of open up and close onto our molecule here, our substrate. And this substrate is actually an arginine, so that's an amino acid. And you can see how that's sort of held inside the mouth here of our Pac-Man or Venus flytrap, like that. And so these would also have, you know, stuff coming off on the bottom here, but this is just showing sort of the mouth part of the Venus flytrap or Pac-Man. To me, it looks more like a Pac-Man in this. But anyway, that is sort of how those structures look. And so when something like sucrose or glucose binds to it, it causes a conformational change that moves down through here and into the interior of the cell, which then activates this Gproteincoupled receptor here. And I will talk a little bit about G-protein coupled receptors sort of in the abstract or more generally here before I go through this particular case. So in this we have the extracellular signal come in, and this is using norepinephrine, which is kind of the model often used when talking about G protein coupled receptors. So that binds to our receptor. There's a conformational change that propagates through here. And so then it activates this trimer here, which is the G protein. And so if we look at that over here, so the G protein, the G alpha here is bound with this GDP, so this guanosine diphosphate. We have our ligand will come here and bind the receptor. That GDP will dissociate, and a GTP, a guanosine triphosphate, will actually bind to that G alpha subunit of our G protein. It causes the dissociation of the G alpha, which then attaches to an effector protein here in the plasma membrane and results in signaling inside the cell, which signals for different biochemical processes to occur. And it's kind of hard to see, maybe if I zoom in a little bit. What you can see here too is that the G alpha is attached to the plasma membrane right there with this sort of fatty acid attached to the protein. And so we have this signaling going on. Then the activation of the GTPase makes this GTP here hydrolyze into GDP. So this little dot here is supposed to be a phosphate. And that actually deactivates the G alpha, and so then the ligand detaches and the G protein reassembles and attaches back to our G protein or our receptor there. And so that's what's going on. So this is the GDP attached. It exchanges that for a GTP, and then it moves over. And in this case, it is showing that is actually cleaving off this inositol from this fatty acid in the membrane. So this IP3 then goes to the endoplasmic reticulum, which then releases the calcium, which then acts as a second messenger for other things. And so I have down here the structures. So we have the PIP2 up here, and that is this phosphatidyl inositol 4,5 bisphosphate. So it's this diacyl glycerol here in blue, and then we have this inositol here, and then when that is detached, we end up with this inositol 1,4,5 triphosphate, or IP3, which goes to the endoplasmic reticulum and results in the calcium release. And I also had the ATP molecule down here. But so that is sort of how this is working in general. And then sort of the specific version here. And so this actually happens. So these T1R2 and T1R3 are also present in the gut, so in the intestine and in the pancreas. And I will talk more about that in future videos when we get to that part of digestion. So I am just going to talk about how this works in the taste buds. And so we have our T1R2, T1R3 receptor here. Some sugar comes and attaches to it. We have our G alpha beta gamma here, which goes and detaches the phosphatidyl inositol here, so the PI45P2, so we end up with the IP3 here. So remember, the IP3 was this inositol 1,4,5 triphosphate, which goes to the endoplasmic reticulum. It causes calcium to move out. So this has the inositol 1,4,5 triphosphate, stimulates the calcium release from the endoplasmic reticulum via the type 3 IP3 receptor. The elevated intracellular calcium activates the TRPM5, which is this channel, leading to membrane depolarization. So there would be an action potential. This shows it as kind of right next to each other, but there'd be an action potential moving down the cell, getting to that channel right there, which then releases the ATP and stimulates. This is efferent. There should be afferent nerve fibers. And so that is how the signaling actually occurs in this. And I actually have a structure here of a G protein coupled receptor. So in green up here is the actual receptor. Then in the sort of salmon color down here is the alpha subunit. The cyan is the beta subunit, and then this purple is the gamma subunit. And this is actually the like I said, the sort of model that we were using, which is the adrenaline receptor. So this is actually the adrenaline receptor here. And so there is this adrenaline sort of analog bound up here in the receptor. Then if we look down here, and you can see there's orange I have overlapping here, because the full structure did not have a GTP bound, but I found another structure that did have that was just the alpha subunit with GTP bound, so I overlapped that with the alpha subunit on here, so we could see this GTP bound in here. We have our magnesium, the gray magnesium over here by our triphosphate. So we can see the GTP bound in our alpha subunit right there. And so that is what a sort of generic, I guess, G protein coupled receptor looks like. So this part would all be inside the cell. This part up here would be outside the cell, where that ligand could bind into there. And so, yeah, that is how the sort of sweet, umami, and bitter receptors work using this G protein coupled receptor mechanism, which is different from the salt and sour, which used the ion channels as receptors. Oops. So we can look back up here. So our T1R1, T1R3 is our umami, our T1R2, T1R3 is our sweet. There are some 30 T2Rs, which are the G protein coupled receptors that sense bitter tastes. And so we have our epithelial sodium channel for our sodium. And then this shows the PKD2L1, but we saw that there was also the OTOP1, which can act as the sort of sour receptors, or they're proton ion channels, but they act as sour receptors. So there was one other thing I wanted to talk about, which was actually the capsaicin. So this is the TRPV1 receptor. We can see that here, and we can see that that is that actually contains several different proteins. So we have the blue, the cyan, the green, and the magenta. So there are 4 proteins involved in that. Then we can see over here, so what this is, so the best known activators of TRPV1 and sort of what its function is for is sensing temperature. So sensing temperature greater than 43 degrees Celsius or 109 degrees Fahrenheit. And what happens is when the temperature is high enough, it opens the pore on here and allows ions to move down through here into the cell, which then once again causes a depolarization and causes an action potential. And I should also mention this receptor is in the neuron. So this is in the afferent neurons. So this is not in the taste receptors. This is actually in the neurons. And there are multiple types of TRPVs. That's why this has a 1 on it. And they all sense temperature, and they're found in other parts of the body than the tongue as well. But the TRPV1 is well known because it's what actually binds with capsaicin. And all the different TRPVs are activated at different temperatures, so they're all temperature receptors. They're all temperature sensors, and they activate at different temperatures. But the TRPV1 in particular activates, it opens the ion channel when it's greater than 43 degrees Celsius, which is 109 degrees Fahrenheit. But other things that can activate it are acidic conditions, which is why acid can taste hot, capsaicin, and also allyl isothiocyanate, which is what is found in mustard and wasabi. And so capsaicin is a specific type of what are called vanilloids. So that's why that vanilloid is there. And we can see some of the places on here, particularly right around here, where the vanilloids can bind to this receptor. And what that does is it actually reduces the temperature at which these channels are opened up, and it reduces it to below body temperature. And so that's why, just your body temperature, the body temperature of your tongue makes you taste heat or feel heat on your tongue. And so that's what capsaicin is actually doing. And then there are these sort of calcium proteins that can actually desensitize this and cause it to close up. But there are also like these protein kinase A's. And so one of the things that the papers talked about is there are a bunch of ways that these things are regulated. So regulated by, the inositol binding, by binding of these calmodulin proteins, by ATP, by protein kinase A, which phosphorylates it. And so we can see that hydrogen binds up here, which activates it as well, and right here. So these things are sort of highly regulated. But the thing that I wanted to point out is that capsaicin binds it, and that reduces the temperature at which the channel is opened up and activated, and so that's why it causes that hot sensation on the tongue when you eat spicy foods. All right, so I think that was everything I wanted to talk about with taste. So I mean, there's a lot of information here, with the salt and sour being the sort of the ion channel sensors, and then the sweet, umami, and bitter being the Gprotein coupled receptors. So those are kind of the things to remember. The other thing to remember is that this is all sort of communicated through these sort of long taste cells like this, so that look like this, through action potentials, which I talked about here, and then releasing the ATP. So when the action potential gets down, it causes the release of ATP, which then binds to these TXP2 and TXP3 receptors on the afferent nerves, which then allows ions like sodium to move through the channels into the cell, causing depolarization and resulting in action potentials in the nerves. But I guess if there's sort of one really big thing to sort of remember from all this, is that this picture of how taste works is untrue. This is sort of a myth I think it was, it's one of those things that was proposed by somebody like a long time ago and kind of just stuck around. But in fact, the different types of taste cells are present in every taste bud here. And so every taste bud can taste the various different kinds of taste. The sort of, big ones being the umami, the sweet, the bitter, the sour, and the salty. That's sort of the big five, as they call it. There's, a lot more to it. So, I mean, just looking at sort of what they have going on right here, there's signaling between each of these three different types of cells using things like GABA and serotonin, and so the different cells are sort of talking to each other, and this is what can kind of result in sort of the complex flavors that we end up tasting. Not we don't just have these sort of five things that are sort of binary, like you're either tasting salty or you're not tasting salty, or you're either tasting sweet or not sweet. So, there's a lot more complexity, and as usual, there are lots of references that you can look at in the description down below if this is something you want to read more into. But this, as usual, is just sort of an introduction to the topic. And I hope you found it interesting and maybe even helpful if this is, you know, something that you're looking to study. But anyway, I don't want to ramble on any longer. I will see you in the next video.